time because we need to get going, okay? Be 50 questions plus there'll be some extra credit questions. And the extra credit questions will be about community. Okay? So, has everybody ever taken a practical where you walked around the room and you wrote stuff down? Yeah? No?
Yeah.
Some people, have you taken practicals where it was online and you write? It's kind of like that. You'll get an answer sheet, looks like this. And then you'll just write, and the practical or application, there's like a few words at the most for an answer. Most of the time it's just one word. Okay? So let's set the rules. The rules are that the allied health programs want it to be timed. So I will comply with that, but not on the first one. On the first one, you can ask all the questions you want during the exam. At the very end of the exam, everybody will be able to ask 2 clarification questions, right? But after that, every practical after that will be timed, so you can only ask questions at the time we're looking at the actual question or that particular topic. And then after that, we're done and we're done. There will be no going back, okay? Because nursing program, dental hygiene program, and the surge tech program want everybody to have time test. So I will comply with that.
Everything is timed in nursing.
Everything is tied to nursing, that's correct. So, you can ask any clarification you want, right? So for instance, if you think that's purple but you're not sure, you can raise your hand and you can say, Pro V, can you tell me what color I'm looking at? That's a clarification question. I'll say you're looking at purple, right? But you can't say things like, Pro V, can you confirm that's gram-positive, right? That's giving the answer away, right? I will give you the look, and then I'll forgive you and I'll just change the question. But if you give a lot of answers away, I might get very grumpy, which is very hard for me to get. It takes a lot to get me grumpy, okay? The other rules are that when you're looking at staining, anytime you see purple or red, you assume it's a gram stain unless I tell you differently. Okay? So you see purple and red, it's a gram stain unless I tell you this is a simple stain. Yep. If you see green and red, it's what? Endospore. Hot pink and blue, it's asbestos. Black and purple, it's capsule stain. Okay? Good? So those are the rules. Okay? Oh, no. I'll get that chair. Here you go. Don't fall. Okay, so there'll be 50 questions, each of them worth 2 points each, and then there'll be 5 extra credit questions. So the total possible you can earn on the practical is 105. Okay? I don't give 2 points for people who study together. I want you to study together, but I have another mechanism and another reason for the way I give EC on practicals. Okay? Pretty good? All right. So let's go through these questions. So identify the gram reaction at the pointer. It is gram positive. It's okay to write gram positive. It's okay to write GP. It is not okay to write G plus. Don't do it. Okay? What's the difference between gram positive and gram negative cell walls?
A gram positive.
There's a whole bunch of things you can do.
Gram positive has a small peptido, or a large peptidoglycan layer.
And gram positive has the same.
Has the gram positive have teichoic acids? Gram negative have LPS. Gram negative have small peptidoglycans. There's a bunch of them.
Thin layer versus
All those are correct, right? I'll give you credit for anything that's correct.
You know, my girlfriend's also microbiome with the professor that's usually next door, and we had an argument because I was telling her, I was like, gram negative has teichoic acids, right? And then it
No, gram
has,
positive have teichoic acids, not gram
I
negative.
mean, the gram positive does, and it also has a thinner peptoglycan layer. She said no, it's thicker, and then the gram
The
positive is
positive?
thinner. Huh?
The positive stuff, I think?
She said that the positive has a thicker one, and the negative has a thinner one, but it has like an outer membrane or an outer layer or whatever.
Gram negs do. Gram negs have an outer layer.
Yeah, and then the peptidoglycan it was like a thinner one.
Thinner, it is.
But we say it's thicker, right?
Gram positives have thicker.
We say gram positive is thicker, but she says gram. Oh wait. I'm contradicting myself now. Yeah.
You know what? You just learn by doing that. I'm okay with that. Let's go on.
I just realized.
Identify the arrangement at the pointer. Streptococcus. You can say coccian chains, that's fine. But,
Is that looks like bacilli.
Eh,
It
okay,
almost
hold
does
on
look
now.
like bacilli.
oh, okay, hold on.
In that point, we
You're
could
right, it does
clarify.
look like it's a coccus. So if you're not sure, this is why we do this.
So,
If
for
you don't
instance-
know, you can raise your hand. Praveen, what kind of shape am I looking at? And I'll say it looks spherical to me. But listen, if nobody clarifies and somebody writes streptobacillus, I'll accept it because it's distorted. Clearly it's distorted, right? Yeah, but they're a cocci. Okay? You can't say spirilli. That would be bad. Okay?
So, is that a rod shape? Is that an acceptable question to ask?
You need to. I'll say something like it looks. I'll put it in a way that doesn't give the answer away.
Okay.
Okay? If you say, is that a rod? Then if I say yes, then I just gave the answer away. If I say no, then now if people know that gram positives are either cocci or rods only, they now have the answer. So I will say something like it looks spherical,
Okay.
and then they have to think about it. Right?
Could you say like, is that an oblong shape?
I would come back and say it's spherical.
Okay.
Okay, so you can ask, as long as you don't give answers away, I'm just gonna answer it in a way that it provides information for you and everybody to answer the question, but I'm not gonna give the answer. Sometimes I will, Christine. So, Marianne, sometimes I just talk and I'm
You
thinking
end
about
up
what I wanna
giving it
say, but I
away.
give the answer away. So it just happens sometimes, and in those times, you just get that for free, okay?
Wait, so you said this is spherical?
Those are spheres. Yeah. Name the counter stain and the gram stain. Safrana. Safrana. Okay? There you go. Here we go. I'm looking at green and red. What are you looking at?
Endospore?
That's the first thing that should pop in your mind. I'm looking at endospore. Anything that's gonna answer here is gonna be about endospore. So, type of staining technique is? Endospore. What's the main function of the green structures?
It's
Survival.
survival.
Identify the primary staining reagent.
Can you say protection?
No,
My
but it's not.
malachite green.
It's survival. Huh?
Malachite green.
Malachite green, very good. What is the decolorizer in this methodology?
Something.
There is none.
Oh.
Somebody said it, I came from somewhere, I don't know where. Water is not a decolorizer, it's a rinsing agent. Okay? And I said that, somebody's gonna tell me the decolorizer is water, and I'm gonna say no, and you're gonna be grumpy at me, but it's not.
There is no decolorizer.
There is no decolorizer.
There was malachite green?
Malachite
The
green is the
main
primary
one.
stain. What's the counter stain?
Something red.
Something red.
It starts with a C, right?
No.
No? Something.
Starts with an S.
Oh,
Safranin.
safranin.
Safranin. All right, here we go. So you're seeing purple, what do you assume? Gram stain, very good. So identify the stain technique. Gram stain. What's the purpose of iodine? Mordant. But there's so many other words that can be used here. You can say it's a fixer, it's an enhancer, it's a binding agent. There's so many things you could write on there that you get credit for, right? I'm more interested in you all communicating that you understand the process than do you fit the exact word that I want, right? Okay? Name the primary morphology seen on the slide.
Coxswain.
Coccus. Coccus. Right?
Are you gonna slam us if we say caucus or cocky?
Well, caucus is singular, coxy is plural. There's a lot of them on
Alright.
here, so if you said coxy, that'd be fine. Okay? I won't slam anybody for no reason. Okay, true or false, the bacterium in this field of view is motile.
False,
Why?
because it doesn't have any flagella.
Because, by definition, coxy are non-motile. What's the decolorizer in this methodology?
Ethyl alcohol?
Acetone alcohol.
Acetone.
You can't just say alcohol, that's wrong. Okay?
Yep.
All right, here we go. So, looky here, what is this method called? The street plate, very good. What's the purpose of this method? Be specific.
To isolate colonies.
Isolation. Isolation. Name the object at the pointer.
Colony.
It's a colony. It is not a round thingy. Yep. What does a CFU?
Colony forming unit.
What if I ask it this way? What does a CFU mean?
It's
One
one
cell.
single
One cell. What tool is used to conduct this method?
inoculating.
The loop. You can say inoculating loop, but if you said loop, that's perfectly fine too. But you can't say that long thing with a round end. No. Okay? All right. Again, you're looking at what color?
It's gram.
It's purple.
Oh,
So you assume,
it's gram
Gram
positive.
okay, gram positive, fine. So what is the gram specificity and morphology? But this time I want you to use 3 letters in this answer.
Gram
GPB or GPR. Either one is fine. Yep.
I have an argument
Okay.
for you. Oh wait, never mind, not this one. Never mind.
Not this one?
There was one, you go back again, that one.
Yeah.
There's nothing on that that picture, that slide, that says that it's a gram stain and not a pink stain.
I know, but the rules are you assume purple or you see purple or red,
You assume
it's a
that.
gram stain, unless I tell you something. This is the Ruth Claudia rule, because she's the first one who brought it up. Okay? Good? All right, here we go. Okay, now looky here. Now, look at what the data's telling you before you answer. What do you appreciate?
It's a flagella.
It's flagella. How many flagella?
2.
Multiple at least one end, right?
Oh.
So now you have the answer. Okay, now let's do it. Identify the staining technique. Is it molar or non-molar? It's a flagellar stain and it is molar. What's the flagellar arrangement? Lophotrichus. Can't be anything else.
So Amphitrachus is only one.
On one end. Now, you could clarify this by asking what?
Does it have multiple
Are there
flagella or
multiple?
is it just one per
Are
each
there,
side?
you can say, am I seeing multiple flagella? And I would say, yes. It's Lophotrichus.
Lophotrichus.
Cause that one on the left, is that 2 at the north pole?
Okay. So I can't tell. But they're the same organism.
So,
But I can't tell from that one. But what gives it away is that up there.
mhm.
Yep. So you have to look at the data. Okay? All right, let's go. All right, this is motility media. Consistency is what? Semi-solid, very good. What tool is used to inoculate the medium?
Inoculating
A needle or an inoculating needle? Very good. Identify which tube illustrates a non-motile organism.
A,
A, why?
because it's
It's only growing at the site of inoculation. Mhm. Very good. What is the percent auger that's associated with the semi-solid medium?
0.5 to
0.5, but if you said 0.5, 0.6, 0.7, or 0.8, those are all correct, because there's a range. We just like 0.5. That's what we use. Okay? Just like in a solid, if I ask what percent auger is in a solid, our preference is 2%. But if you said 1.5, 1.6, 1.7, 1.8, 1.9, or 2, those are all correct, because there's a range.
Can we say 50% or is that?
No.
So it's
Because 50% auger is a lot.
not,
That would not, yeah,
okay.
that wouldn't work. Okay?
So that's 0.5.
Okay, so here's a microscope. And I'm not gonna ask you every single thing. The ones that are labeled here are probably the ones I'm gonna really focus in on, right? Including, but they're not labeled, or the course, and the fine adjustment, and then the diaphragm and the condenser. Right? But let's just answer what we got here. Identify the part of the microscope that's labeled 1.
Eyepiece.
So it's the ocular or the eyepiece. Okay? What is the magnification?
10,
10x.
10, 10.
In 10.
Or times 10.
So 1 also has another function. What's that other function?
To
It's an
visually see
eyepiece.
stuff.
Huh?
To visually see.
Brings the field of
stuff.
view to eye. That's right. Identify structure labeled 4. It's right there.
It's nose piece.
Nose piece. What is the magnification when using all immersion objectives?
100.
100 times. But what's the total magnification if you're using the oil immersion?
1,000.
What's the purpose of oil? Corrects the refraction index.
So like in question 27, it says when using the oil immersion lens, that like implies that we're looking through both the.
It does imply, so if you said 1,000, I'd probably give it to you. Yeah. Okay? If the question's ambiguous and I say it that way, then I'll be very open to giving you things that you interpret. Okay? Anybody else? Okay. So here's a plate that we did kind of on the first day of class. Do you remember? So what's the origin of the structure labeled A? Water. It's glistening, right? What's the origin of the structure labeled B?
Dirt.
Soil. It's very rough looking. What about this one here?
Which one? Human.
Humans, people, animals. Yep, all those are correct. Okay, here we go. How many different species are likely on this plate?
Is it? Would it be 5 or 4? Because
Let's count them. First of
other
all,
colony on the outside.
let's
has
count
a
them. First of all,
regular-
this one, this one, this one, this one, and that one are the same. That's 1, 2, 3, 4, 5, 6. So I would accept 5, 6. Yep. Good?
Question though. Because even if like the one on the furthest
This one?
right, that one
So
has
if-
more of an
if-
irregular shape.
If you told me, Provine, because that one looks so different than the other guys, I thought it was another one, and you said 7, I would give it to you. But there has to be, it has to be that the argument came from the fact that you were analyzing this and you analyzed it different than I did. But if you tell me that story and I can see why you got there, I'll give it to you. To me, this is, you're learning, you're analyzing things that are not easy. I'm gonna be pretty open to you telling me, all right, now you can't just say, Shane told me, then I'm gonna go, that's not gonna be good. He's gotta have a really good argument. Yep.
So if I said there were 5,
Uh-huh.
and there was 1,
Mm-hmm.
2,
Mm-hmm.
3,
Mm-hmm.
1, 2, 4, 5.
Yep.
What am I leaving out?
The little guy, right there.
How do I know that's not an artifact?
It is not an artifact. That is a colony.
So this would also be a colony down here?
Yes, but like
That would be a
that
colony?
one. Yep. Yep.
That would be a
So
colony?
would
Yep.
you tell us that there are no artifacts?
But there are no artifacts on here that I can see.
Okay. So that little tiny guy is not a blurry picture, it's a full colony.
Colony.
So how many you said there were 6?
But we wanted the colonies within the colonies.
There is a colony in the colony. That is correct. So I'm counting them. Here we go again. 1, and then these guys are all the same. 2, and then these little guys are all the same. 3, 4, 5, 6. That's what I get. But Sarah said, there's another one. This one looks funny. It looks different. I can see why she would say that.
And I counted that one separately.
You counted that one separately also? So, okay, you get another one, that's fine.
How do we know that the one inside isn't just, is it because it's a different size than the,
Mhm.
okay.
But if you said Pro V, if you said Pro V, when I was analyzing this, I thought that was artifactual. then I'm like, okay, you have a really good reason for why you have a different number than I do. You're gonna get that, you're gonna get it right. I'm not here as an obstacle. I'm not here to be the guy who just makes your day terrible, right? I'm more interested in you learning, okay? Because there's nothing black or white about this stuff. It's all based on your judgment, okay?
So
Good.
you're gonna accept anything in the range of 4 to 7?
Yeah,
Okay.
that's fair.
Yeah.
And you don't need to write a dissertation. You don't have to say, I selected 7 because I think that this one out here is really different. And you don't have to say that, right? Just, you'll get an opportunity to tell me why. Okay? Good?
Just don't put 1.
Don't put 1, that would be wrong, right? Okay, here we go. So this is just broth. Notice I said it's just broth. So what things can you not tell me? You can't tell me it's an aerobe,
Oh.
you can't tell from here. Okay, so now let's answer. Name the term that is used to use for the growth pattern seen in 2A, and that would be what?
Turbidity.
Turbidity. I will not accept cloudy. Has to be turbidity. Okay? And then what is the term used for the growth pattern seen in E? This one right here.
Pellicle.
Pellicle's good. Can you say something else? Some people might say that's sediment,
Yeah.
and I would be okay with that.
On E?
Mm-hmm.
Very slight sediment.
Very, but it's there.
So would you want both or one or the other?
One. I'm really looking for pellicle. But there's gonna be somebody in here, I'm not gonna mention your name, Sarah, but who's gonna come up with a different answer, because that's just the way she is. She likes to analyze. She overanalyzes things. Did you see how much trouble she had giving me a number? She was trying to figure out what number would best fit. Good? Any questions? Here we go. So what am I looking at? A negative stain. So the answer to the first one is negative, right? What's the advantage of doing this?
You can see
No
the
distortion.
morphology.
Is the dye used in this acidic, basic, or neutral? Acidic. Very good. What's the purpose of a negative stain? I wouldn't say that because that gives the answer. What's the purpose of the stain? To tell morphology. That's it. Okay? Okay, what is this thing called? Anaerobic jar. Right, this morning somebody tried to get cute on me and said, it's a gas pack. I'm like, no, that's this little thing right here. That's not the entire thing, right? When set up correctly, this is an anaerobic jar. When set up correctly, does this container contain CO2?
Yes.
Yeah.
No.
No.
It does not. Water. Contains water. The candle jar does. And I might throw a candle jar in here. It's not here to now, but I might throw it in there, right? So, but that would increase the CO2, decrease O2.
Makes water.
Water. So, what type of bacteria would grow in this container? There are 3 possible choices here. What are they?
Arrow.
Facultatively anaerobic, that's correct. Anaerobe, that's correct. One more. Aerotol. Who said that? Coming from the back of the Kimmy. Yeah. Alright. But if you said microaerophile, it's incorrect. If you said aerobe, it's incorrect. They won't grow in here. Okay? Good?
What do we put as anaerobic?
What's that?
What do we put as anaerobic?
And just anaerobic? That's fine. That's perfect. To me, obligate, anaerobe, and anaerobe are the same thing. Okay? All right. True or false? An aerobe will grow in this container. Say it out loud, Donna, like you know it. Don't whisper it like only somebody real close to you, even closer than Daniel is, can hear. He didn't hear you. False. Right. Okay, here we go. So what type of stain is this? Acid fast, very good. The blue things represent what? Non-acid fast cells, right? What is the decolorizer in this methodology? Acid alcohol. Very good. What is in the actual cell wall of the fuchsia bacteria that make it resistant to staining and contribute to its virulence.
You said the sugar's naganam.
Not naganam, mycolic acid. Mycolic acid. All right. So here, this is thioglycolate. Now, what are the words you're going to throw at me? Aerobic, microphilic, facultatively anaerobic, aerotolerant, and anaerobic. So what? Identify the type of bacteria based on the oxygen in C. It's only growing at the bottom. No, it's only growing at the bottom. It's an anaerobe, right? Identify the type of bacterium in D. Only grow at the top. Aerobe. Good. Anybody have any questions? There'll be some safety questions also. Ma'am?
What's student A and B?
So A is the control, and B is a facultatively anaerobic organism.
It's everywhere, right?
It was terrible. I thought it was ambiguous, so I didn't ask. I tried it in the morning class, but then they got all like, I'm like, fine. Yep.
I have a question about something that was on my practical that I have not heard you mention in this class.
Artifact.
Nope,
What?
not that. And I didn't hear you mention it in my class either. Tray colors, there's the blue tray, which goes in the fridge.
That's a fair question.
And there's the red tray that goes
Which
in the
goes
heat.
in the
And then there's
incubator.
another tray. And I have not seen the other tray. I went and looked for it today, could not find it.
It's
What
green.
is that
It
other
is
tray and
green,
what does it
yeah.
do?
It's green and it grows.
It's not even yellow, sorry y'all.
It's green. It grows fungi, and it goes in the 25. I won't ask that one because I haven't talked about it. But the
You
blue,
asked that in mine.
the blue and the I know because you all ask more questions than this class has yet. I think some people in here are still thinking I don't have their best interests at heart. So I blue and red are perfectly fair,
Okay.
right? Permissive temperature is perfectly fair. What's permissive temperature? Okay, you weren't listening second day of class. Body temperature. Right. What BSL level is the lab at? You weren't listening first day of class. 2. Where's the emergency shutoff valve?
And the
Well, back where? By the door. Very good. What paper do you use to clean objectives? No, not me. Like you're playing -10. Lens paper. What type of paper do you use to take off extra water from slides you've made? Bibulous. Very good.
Wait,
What's the disinfectant used in the laboratory? By identity and concentration?
10% light.
10% Lysol. Very good. All fair.
Is that like on PowerPoint anywhere?
No.
Yeah.
Okay.
It's also in that sheet I gave you first day of class. I said, you probably should know these. You remember that?
I think so.
So is the skull toxic?
Is what?
The skull?
Poison.
Poison?
Poison.
Biohazard, heat.
Yeah, oh, the other thing is, what I like to ask is, on the hazardous communication, you should know what red, blue, and yellow mean. What does red mean? Fire. What does blue mean? Health hazard or toxicity. Yellow,
Reactive.
reactivity. Okay? So you'll probably get 6 of those because I have to do at least 10% safety. Or the other thing is I have to do a weekly safety exam. I'm just not gonna do that. So I just incorporate safety into each of the lab practice, except the last one, Ruth. I don't do it on the last one. Why? Because you're leaving me at that point. So you're gonna be somebody else's issue at that point if you make troubles. Okay? Anybody have any questions? I promise you, you will not hear anything we haven't talked about in class. But sometimes you'll hear things that you think you might not have heard in class, but other people were listening and they heard it.
That green tray was out of pocket.
3 people got it right.
3 people.
3, wow. That's a lot.
That means 3 people were listening. You should know everybody in this class.
Really?
I know
Really?
like half
Because
of them.
if I remember correctly, there was some discussion that
That what?
That some people couldn't remember other people's names. I'm just saying.
I know everybody's name in here.
I know everyone.
I'm really bad with names. What are you talking
Really?
about?
Well, then you might not get the
I
extra
don't know
credit.
everyone's
You knew
name.
our names like on the
But I
first
don't
day.
need
Yeah.
any credit.
Who is not in the
I
group
passed the
me?
class already.
Just join that now.
Oh yeah, we can look at yeah. It's
And
Is
if you-
everyone
if you
on there,
If
though?
you told me something about you, and as long as I get your permission, I might use that, right? So I might say something like, which one of our colleagues in here is from New York?
Who's from
And I would
New York?
expect you to be able to tell me their name. The problem is what? You don't know who that is, so I'll say, will the person from New York please stand up? Please stand up.
I'm also from.
Okay, well then
There
you
you
can
go.
stand up too. But you haven't told me that, so I don't know that about you, right? Who in this room is dating an individual in my colleague's class?
that guy.
Yeah.
Wait,
Right.
what's that guy's name though?
Yeah, that's right.
Josiah.
You gotta know his name,
Josiah.
right?
So
Good.
like you-
Who in this room went to Amarillo on the weekend of the Kansas City win, the big win, right? So I remember everything you tell me because that's how I know you, right? Who in this room was my model and they were the hydrogen and NAD picked them up and took them around the room? What's her name?
What's your name?
Was it Dan?
You don't know.
Eva.
Yvonne. If I know all 22 of you, you should know everybody too. Because the most important individuals or the most important things of this class are the people in it.
Alright, everybody update there.
You don't get to know them, but then you don't get the EC. It's extra credit.
Can we get like a
Oh,
little
thank
paper
God. You can
do whatever you
with
want.
people's names?
But I am through with the review. Does anybody have any other questions? That is a pretty thorough review. I can't really deviate from those questions very much, but I'll find a way to be creative with them.
How about we all go around the classroom, stand up and be like, hey, this is my name and this is how this does.
You can do that. Let me sit down.
Let's
Let
do
me-
it right
This
now.
is how you spell
Right
it.
now.
Yeah, and we all like record it.
Is spelling
That'd be
matter?
the best
Yes.
way.
For
Yeah,
names,
spelling will
yes.
matter for names.
What?
Hey, don't give him new info that he doesn't know about you yet.
He didn't tell us that, and it did count. I spelled his name wrong.
Yeah.
The last
Oh, does
name?
spelling matter? On the
Cause
names?
On the
two
names, yeah.
two L's, two R's.
For the names, because it's extra credit.
Oh, okay, alright.
But I don't, listen.
VI.
Nobody's recording me, right? Fatima, stop it
No,
now.
I'm taking a picture, I'm not.
Okay.
V-I-L.
I do not count off for spelling on the practical. Because I'm dyslexic and I have a hard time with spelling. I've had a hard time with spelling all my life. But I always fought through it. But I have a big piece of my heart that goes to anybody who has problems with spelling. Therefore, I don't really count off for spelling. If it's phonetically correct, I'll give it to you. But if you mean cat and you say dog, then it's wrong. But if you mean cat and you spell it K-A-T, that's okay. But if you mean cat and you spell it P-I-G, that's not right. Yes? All right. Y'all have a great weekend. I'll see you on Tuesday.
Right. Are we gonna do the name thing?
Are we gonna? Sarah wants to know if we're doing the name thing.
Okay, okay.
Hey, someone take a video, right?
I got it. Okay, you start us off, Josiah.
Alright, my name is Josiah. The obvious fact is what he said earlier, but my girlfriend
this is for
in the
the
room next door. And then, does anybody know how it's spelled? Or does anybody know how Josiah is spelled?
How do you spell it?
That's Josiah. Alright.
Okay.
I'm Christine. It's Christine with a K.
I don't get
Spelled
this.
like Kristen, but with an E at the end. And my fact was I have a bachelor's in sociology.
Okay.
I'm Yvonne, it's Y-V-O-N-N-E.
But not for life.
Right. I didn't say effect on the first two.
We can use the fact that he did the hydrogen thing with
Oh,
you.
okay,
Yeah.
Oh no, there's other things you don't know about her. Like she's very cute when she does it. She goes.
When she was the hydrogen
Yeah,
in
yeah.
the. My name is Ruth, R-U-T-H. I'm going through a messy divorce.
And you're a grandma.
But I would never say something like that, okay? I would never say something like that.
That's why she used this. so
Do
she
the tattoo
can-
one. That's the only thing I might use.
Yeah.
I'm Rebecca, REBECCA, and I went to Amarillo for the Super Bowl weekend. For a baby shower.
So, just a quick question, I don't remember what I said the first day, I just kinda like threw things out
Yeah.
there.
But I know a lot about you.
Okay. I'm Fatima, I took him like twice already, I'm going for nursing, I already have a degree in surgical technology.
Okay, people are like, she took it 3 times. She was really very good the whole time she's been with me.
It just expired, cause you know, like
There you go.
science is
I
expired.
want,
That's
I
all.
want to put some context around that, because I don't want people to think anything good about Fatima as I do. I think she's wonderful.
Anything bad, thank you. Just let anyone think
Yeah.
anything
I
bad.
haven't had enough coffee.
Any publicity is good publicity.
Okay.
At this point.
My name's Daniela with 1 L. Because usually you spell it with 2, but it's 1 L. It's called that. I didn't say anything about myself.
I'm Ariana, spelled with 2 N's, and I like to crochet.
And she loves K-pop.
Oh, yes.
So you don't think I'm not paying, I'm paying attention to everything that I see, everything that I ask. I know you all better than you think.
My name is Vivian. I'm going for dental hygiene, and I'm a mother of 2.
How do you spell that?
Vivian. Really simple, classic.
My name is Donna, I'm gonna be working in Colorado this summer.
I'm Daniel, Daniel, and I'm a Dodgers fan. That's about it.
She's also from California.
I am from California,
Oh, man.
Paris, California.
Don't give me more information. Just California. Less is more.
I know a lot about people that y'all don't think I know, right?
California's dope, bro.
Okay, my name is Sun, S-U-N. I think I told you guys first time, because I just turned my 60th birthday.
Shane, S-H-A-N-E, and I'm from New York. I think that's what we'll stick with that.
There's other things about him that I know.
Oh dear God.
My name is Anna, 1 N, and I live in Bastrop too. I don't think he knows much about me either, so.
Okay, my name's Kimberly, there's no E, it's just L-Y, and I disclose no information with him.
Don't you have that sister?
Oh.
Yeah, there's
I
a
have a
lot I know
sister.
about you that you don't have any idea.
Yeah,
It's
she's
just that you
I
see my
have
talk
a
a lot.
sister that took him, that's it.
That's it.
My name is Jasmine with the S and an E at the end, and I have 3 cats.
My name's Alyssa, it's spelled A-L-Y-S-S-A. I'm from Salt Lake City, Utah.
You went to Mardi Gras?
I went to Mardi Gras. I have a serotonin molecule on my arm. I think you could point that out. That's it.
My name is Brittany, spelled B-R-I-T-T-A-N-Y, and there's nothing for me.
My name is Susana. I am from Mexico. That's it.
Let's see if I can do it.
Could you spell your name for us? I'm not giving you
My
any.
name S-U-S-A-N-A.
I'm Sarah. Sorry.
Without an H.
I'm Sarah. Without an H, yes. I do not do H's.
I
Stuff.
know a lot about her.
Yeah, he knows a lot about me, so I don't know, man.
The one with the most information will be Sarah.
Yeah.
That's all, but but remember,
Spell my name, that's your figuring too.
Oh, how do you spell your name?
M-E-Y-E-R,
like the lemon.
space,
Oh, there's a space? I thought.
A-N-N-E. E at the end.
Oh, wow.
Fire like the lemon, space, and with an E.
She plays golf.
I do a lot of things. I'm an Eagles fan, that's my most interesting trait.
Yeah.
I can get out, cut out
Go
the
Birds.
other video.
Did you say who you were?
Yeah, everyone
Like,
has.
I did.
Oh, okay.
Yeah, I was-